you can see like in this particular example, MDM2, for example, is showing high expression in the cancer data, also having quite good expression in the normal data. So we need to look for those E3 ligase that can having high expression in the tumor and low expression in the normal data set. These are some more examples. So in this particular example, you can see CCNF is one of the E3 ligase, which is showing high expression in the cancer data and very low expression in the normal data. So this can also be a potential E3 ligase that we can use after comparing with the other factors that we are doing the study to find the potential E3 ligase. Now coming to the next part, what we are trying to see also, we are also trying to see across the different cancer type. So we have also plotted the box plot and the dot plot to see the expression profile of couple of E3 ligase across different cancer type. And we try to see how the expression pattern is changing across the different cancer data. And can we use this particular E3 ligase for any of the cancer type? So we can create a different bucket for different cancer type, and we will have a list of E3 ligase that we can use for different cancer therapy. So for this example, let's say MDM2 is showing high expression in these cancer type, let's say breast cancer and other couple of cancer type, but also showing some expression in the normal. But if you're going to compare for this last thymus cancer type, so they are showing high expression in the cancer data and having low expression in the normal. So this can be MDM2 can be used for this particular cancer type if they are going to target for particular substrate or any protein of interest. And after collecting this information, basically we try to look for, so based on different E3 ligases and their specific substrate, so E3 ligase can have many different functions. So we can see like they can be the part of proliferation and apoptosis. They can also play a role in the DNA damage repair. They can also play a role in the immunity and the metabolism thing. So for example, if we take this example, so this is the plot which is taken from some research article. So for example, we are focusing on one of the E3 ligase, that is HACT3. So basically, this particular E3 ligase playing a function in regulating the cancer cell progression. So now after collecting these information, we have to look for the E3 ligase and the substrate interaction, basically finding the protein-protein interaction. So to find the protein-protein interaction, we try to look for use for a couple of database. So we have used the UV browser, BioGrid, and String database to look for the protein-protein interaction between E3 ligase and the substrate and try to find the known and the predicted substrate for the couple of E3 ligase. So this is the example of UV browser. So on the left-hand side, you can see there's a E3 ligase HACTD3. And the plot basically, this is like whatever the colors we are seeing in the red color, all the 6 substrate, these are basically the known substrate for the E3 ligase, and we have a couple of predicted substrate for the E3 ligase that was predicted based on some literature. So UV browser basically using a 5 database. So this particular UV browser is using a database kind of thing. So they are using the manual curation, protein go annotation, protein domain, and couple of other things like neural topology kind of stuff to predict the substrate for the E3 ligase, and also going to provide the confidence score and couple of evidence that is going to support the interaction or the protein-protein interaction, like the E3 ligase versus the substrate. So we try to find the known versus also the number of predicted substrate for each E3 ligase that we have curated. Then what we try to look for a list of E3 ligase having a less number of interaction with a substrate. So let's say, for example, in this particular example, so we try to check for the expression profile of 2E3 ligase. So we try to look for one of the E3 ligase CCNF and AFMR. So we try to look for the expression profile the protein-protein interaction of these 2 E3 ligase, and we try to look for the number of known substrate and also try to look for the predicted substrate. So you can see like for this particular E3 ligase, there was only 3 substrate that is coming as a known, and other are the predicted one based on the score. So we can use based on the score, like which one we can use, or which this particular E3 ligase can be used to target these specific substrate kind of thing. Now we try to look for one more important thing is, so we need some specificity. So in this particular example, let's say on the left hand side, I have an E3 ligase. We try to look for the protein-protein interaction. So what we found is basically this particular E3 ligase has, let's say, four known prediction, like four known substrate and having a one predicted substrate. So you can see this particular E3 ligase has showing interaction or the protein-protein interaction with a couple of, or you can say few substrate molecule. But same if you're going to see on the right hand side. So this particular E3 ligase, hectid E3, having a couple of substrate. So if you are going to use this particular E3 ligase, they can interact with other substrate molecules. But if you're going to use this particular E3 ligase, they are going to target and having the specificity because of having less number of interaction or having less number of substrate they are going to interact with. So we are looking and preparing and targeting for those E3 ligase that is going to having the high expression in the tumor and having the low expression in the normal data set. Second thing is based on the other factors like whether the ligandability is available, whether the structure is available. And then coming to the protein-protein interaction, we are trying to look for E3 ligase that we have curated. Also, we are checking the protein-protein interaction to see whether that particular E3 ligase has more number of substrate or more number of interaction with the other substrate, or do we have a very few substrate that is going to interact with that particular E3 ligase. Then after that, we try to look for, so we try to look for other E3 ligase. So we found one interesting thing. So this is the one E3 ligase C10ORF19 that is coming in the list curation. So we try to look for the other profile. And also when we check the protein-protein interaction for this particular E3 ligase, you can easily see the number of substrate for this particular E3 ligase is only 6. And also this particular E3 ligase and the one of the substrate molecule for this particular E3 ligase is TP53. So TP53 a major cancer driven factor. So we can say, for example, E3 ligase, this particular E3 ligase can be used for the TP53. So what we try to do, we look for this particular substrate molecules and try to look to find whether any existing E3 ligase that is using in the PROTAC system to degrade this TP53 molecule. Then we try to find and we get to know there are a couple of E3 ligase that is used in the PROTAC system like VHL and other kind of thing. So that one of the substrate for that particular E3 ligase is also TP53. So and you can also see this particular E3 ligase C10ORF19, which interact with less than 10 proteins or the substrate, including the TP53, which is a major cancer driven factor. So the current E3 ligase that is interacting with TP53, such as VHL and other E3 ligase like DCF11. So that is going to interact with more than 200 substrate or targets. So which may induce the reaction with other targets, or we can say we have the unexpected targets. And thus what we can say like this particular E3 ligase C10ORF19 may be the superior candidate to serve as a TP53 specific E3 ligase. So instead of using this VHL for targeting the TP53 molecules using the PROTAC system, because there they are going to interact with a couple of other substrate. So to make this substrate specific and like having the less interaction or minimizing the off-target effect, we can use this particular E3 ligase as a potential E3 ligase in the TPD system, in the PROTAC system to target the TP53 molecule. Now, coming to wrapping up the take-home thing, what we can do and what we have learned so far. So basically the specificity. So specificity ensures that only the protein that we are targeting is going to be degraded. So we have to minimize